Good evening, everybody. My name is Taryn Hawkins. This is a presentation over cell cycle regulation, and we will be walking through it first, starting with our objectives. Our objectives are to be able to identify and describe the stages of the cell cycle, including interphase, G1, S, G2, and the mitotic phase, which is when mitosis and cytokinesis come into play. We're going to talk about each of these phases a little more in depth to talk about how significant of a role each phase plays. That way we can better understand why cell cycle regulation is so important. Our next objective is to explain the function and significance of cell cycle checkpoints, G1, G2, and M, in maintaining genomic stability and preventing the uncontrolled cell proliferation. The next thing we are going to talk about is identifying key regulatory proteins. These proteins will play an important part in cell regulation, and we are going to identify them and talk about them. And the last thing we will talk about is we're going to analyze how mutations or dysregulation in the cell cycle regulators can lead to disease such as cancer and explain the molecular mechanisms behind these processes. So what we are doing is we're going to start here with the phases of the cell cycle. We've talked about this before, but we are going to dive in a little deeper today. So the cell cycle is an ordered series of events that involve cell growth and division that produces two new daughter cells. So the cell cycle is an ordered series of events that involve cell growth and division that produces two new daughter cells. The cells that are going through the process of cell division proceed through a series of events that are precisely timed and carefully regulated. Without this, cells can go awry, and we will see that in this lesson. These stages include growth, DNA replication, distribution of duplicated chromosomes to daughter cells, and division. These cells have to grow big enough to be split into two, and they have to duplicate that DNA because they want each new cell to have a copy of the DNA. Once the cell is divided, two identical daughter cells are produced. There are two major phases in the cell cycle, and they are the interphase and mitotic phase. We will just start our discussion with interphase. So we're starting at the beginning. Cell growth is steady. It's going at a steady rate during interphase. Think of a kid. Between growth spurts, they are growing at a nice steady rate. Most cells double in size between one mitosis and the next. They need to be big enough to split into two. Interphase is important because during this time, the chromosomes are decondensed and distributed throughout the nucleus. So, when you look at these, this is a nucleus that's fixing to go through mitosis. These chromosomes are paired up, but before it actually goes through splitting, we're going to be over here at the beginning. These chromosomes are decondensed. They're moving to either side of the cell. They're getting ready to relocate. During this time, the cell is metabolically active and thus is continually growing. DNA is not being replicated at this time. It's just decondensing, not replicating. The G1 phase is then followed by the S phase. The S stands for synthesis, and this is the time when the DNA replication is taking place. So during this time, we're making copies. So that when we get here, we have enough copies to be split in between two cells. G2 phase is another gap phase. During this time, the cell is continuing to grow, and proteins are synthesized in preparation for mitosis. We talked about that there's proteins that are necessary for mitosis. Well, during this time, these proteins are being synthesized so that mitosis can happen. Once the cell is ready, it will enter the M phase. This is when mitosis actually happens. This is when the cells actually go through the steps and end with cytokinesis. So let's take a deeper look into the phases of mitosis. At the beginning, we have interphase. It is the longest. This is when the cell is growing and getting ready to go through mitosis. After interphase, prophase will be the first stage of mitosis. During prophase, the chromatin fibers condense into chromosomes. So this is when they kind of condense down, they tighten up, they come together. These replicated chromosomes appear as two identical sister chromatids that are joined in the middle, which is what is called the centromere. So these are sister chromatids that have come together, and right here in the middle is our centromere. This is going to play a big role in cell cycle here in a moment. At this point, the mitotic spindle is also beginning to form the centrosomes, and the centrosomes begin to move to the opposite poles of the cell. They are being propelled by the lengthening microtubules between them. There is also a stage between prophase and metaphase called prometaphase. So during prometaphase, this is when the nuclear envelope starts to fall apart. It's important for this nuclear envelope to fall apart in order for these chromosomes to move from one end of the cell, one side of the cell, to the other. If that nuclear membrane was in the way, the chromosomes couldn't move, each cell would not get the correct number of chromosomes. When this happens, the microtubules can come into the nuclear area and bind to the chromosomes. This happens at the kinetochore, which are specialized protein structures that are found at the centromere. So right in here we have kinetochores. These are what our microtubules attach to start pulling these chromosomes apart. The microtubules will play a large role in the next phase, which is metaphase. During metaphase, the chromosomes have lined up at the metaphase plate. That is in the middle of the cell. It's not necessarily an actual plate. It's just called that because they all line up in what looks like a nice straight little line. All the chromosomes are attached to microtubules at this point through their kinetochores. They're ready. They're ready for the next phase, which is anaphase. And in anaphase, this is the shortest phase of mitosis. This is when it happens and it's, they're over the hill, the big stuff has happened. This is when the sister chromatids break apart and begin moving to opposite sides of the cell. So we've gone from a pair to homologous chromosomes. Okay? By the end of this phase, two halves of the cell have the same number of chromosomes on each side. So we have two cells that each have the same number of chromosomes, but they're still one big cell, but they're starting to pinch down in the middle. This is when telophase and cytokinesis come in. During this time, two daughter nuclei form. So around those chromosomes that got split between the cell, a new nucleus is going to form around them. This allows these chromosomes the chromosomes will decondense and they'll relax. And when this happens, mitosis is officially completed, and the last part that is left is cytokinesis. And this is when the cell splits into two, and we have two brand new daughter cells. Now, why is it important that during that process, during this replication, during the separating of the chromosomes, why is it important that we have these checkpoints and regulation? They're essential to the continuation of the process. If the conditions are not met, mitosis will stop. So if everything's not perfect, mitosis will stop. That is a regulatory process of keeping bad cells from continuing to go through mitosis and proliferate. If checkpoints are bypassed, certain pathologies like cancer can occur. So the first checkpoint we're going to talk about is the G1S checkpoint. It's also known as the restriction checkpoint, where things like growth factors, DNA damage, cell size, and nutrition are influenced. So if the right growth factors aren't there, if the DNA is damaged, if the cell's not quite big enough and it reaches this checkpoint, it's going to stop going through mitosis. It's going to say, halt the process, we are not ready. However, if it passes the test, it will move on to the G2/M checkpoint. This is known as the DNA replication checkpoint, and it is influenced by improper DNA replication or DNA damage. If it hits to this point and the DNA hasn't been replicated properly, or if there's damage, a mutation, a deletion, something along those lines, mitosis will stop. The final checkpoint is the metaphase anaphase checkpoint, because this is when that cell is fixing to split into two, and the DNA is going to be moved and 2 brand new cells are going to carry on that DNA. So in order for a cell to pass this checkpoint, the point is regulated by the attachment of the mitotic spindle to all chromosomes. If there is no attachment, mitosis will not proceed because those chromosomes won't be split and put into 2 brand new daughter cells. So I mentioned earlier that there are protein players that are involved in the cell cycle, and let's take a look at some of these. Now, these are not all of them, but these are crucial ones. There are 2 that I haven't listed that play just as important part, and I want to talk about those real quick. The first one is the mitotic spindle protein, and it's a machinery that uses microtubule-based motor proteins to organize and drive cell division. So these particular proteins really get things going and make sure things go the way they're supposed to. The next one is kinesin-like proteins that regulate the assembly of the bipolar spindle as a critical point that is necessary for the initiation of mitosis. The next is a checkpoint that is called the spindle checkpoint. As there are many proteins involved in the spindle checkpoint, those could be targets for cancer treatment compounds. So we've gone over those. Let's talk about the ones we have on my list here. The first one being CDK, cyclin-dependent kinase, or CDK1. It is a protein kinase that plays a big role in regulating cell cycle as it drives a cell into mitosis. So this is the protein that gets the ball rolling. Now, it doesn't get the ball rolling and it leaves. It sticks around for a while to make sure that the cell finishes the cycle of mitosis. CDK1 is also expressed in many tissues and has numerous other functions like initiation of the nuclear membrane disassembly during mitosis. So while it gets the process of mitosis started, it's also a key player in making sure that nuclear membrane breaks down. And we know that's important because if that nuclear membrane doesn't break down, the DNA can't be split into 2 new cells. The next protein on the list are called M cyclins. These proteins are found at the start of S phase and then reach their peak to signal the transition from G2 phase to the M phase. So how it works is it does this by binding to CDKs and forming complexes that are used to trigger the M phase. So it and CDK get together and they form a complex that goes, hey, it is time to start mitosis. M cyclin also helps promote events such as chromosome condensation and nuclear envelope dissolving. So as we've seen, these proteins are not one-hit wonders. They play many parts in the process of mitosis, so they're all very, very important. Next up are protein phosphatase. They control mitotic events such as mitotic entry, spindle assembly, chromosome architecture, and mitotic exit. These are enzymes that remove phosphate groups from proteins, thus regulating their function in many cellular processes. As they are structurally and functionally diverse, they have been divided into 2 families that are based on their substrate specificity. Much like other proteins, research into them has led to insights into therapeutic strategies for treating some diseases and cancer immunotherapy. So these enzymes are important. They play a whole big role in mitosis, but they also have many other roles around the body. The next one is DNA helicase, and I put an image here because it is the one that reminds me of the first time I heard about it when my professor talked about how it's the zipper that unzips the double helix when it's time to go through transcription and translation. But the DNA helix protein plays a role in more than just mitosis. They are found in genetic recombination, transcription, replication, and repair. So again, these are multi-player proteins. They do a lot. It is also the enzyme that separates the double-stranded DNA into single-stranded in replication. However, during mitosis, this protein is involved in allowing sister chromatids to separate. So while, when it's working with DNA, yes, it's unzipping, but in mitosis, it helps those 2 sister chromatids separate so that they can move to the separate sides of the cell and, make the two daughter cells. The final protein we will cover is polo-1-like kinase. Polo-like kinase 1, otherwise known as PLK1. This is an important regulator of mitosis and is a serine threonine protein kinase and is essential for centrosome maturation, mitotic chromosome segregation, cytokinesis, and DNA damage response. As it is highly expressed during mitosis, elevated levels are found in some different types of cancer. We keep going back to cancer. We keep kind of talking about cancer. We're going to better understand why I'm going over this as this presentation goes on. The next protein I really want to hit on is P53. Now, this is a pretty impressive protein. It's a tumor suppressing protein. It is a DNA binding protein and a gene regulating protein that detects anomalies. So it can see when something's going on. This protein is normally bound to an active MDM2 protein, and when they are not bound together, P53 is not activated. So right here, it's not activated. It's not doing anything but chilling. In order for it to become activated, P53 must separate from MDM2, and they must remain separated for P53 to act. So how does this happen? So something, DNA damage, cell cycle abnormalities, something is triggered, and cells that are dividing can be put under, these are chemical or physical stresses that when it happens, ATM kinase is activated. And what ATM kinase does is it phosphorylates MDM2, which causes it to dissociate from P53. Once it's dissociated, the P53 is free to do its job. So once these two are split up, P53 can go in and say, hey, is this something we can repair and restart, or is this something where apoptosis needs to happen? And by having this protein do this, it helps with cellular and genetic stability. It is important for cellular and genetic stability to have that protein kind of walking around and making sure things are doing what they're supposed to be doing. Because, there are consequences of things if the cell cycle doesn't go through the way it's supposed to. And let's talk about these consequences. So the GO phase, we saw this earlier in the image, it's also known as the resting phase. This is when the cell is kind of done working, it's retired, it's waiting for, the final time. During this time, the cell is performing its basic functions and regulatory processes. However, it's not dividing. It's just living its life. Cells will go through the cell cycle until it reaches the GO phase, and then they become terminally differentiated cells. They no longer need to go through mitosis. They're done. So when this happens, they are usually cleared by apoptosis. However, there are anomalies that can trigger a GO phase cell to begin cycling again. When this happens, these cells that are not detected by apoptotic defense mechanisms and can undergo uncontrolled cell divisions, leading them to become cancer cells. Again, the word cancer has come up again. So I think we're having a pattern here of when something goes wrong in the cell cycle, one of the consequences is cancer. So let's talk about cancer. What is cancer? Cancer is the result of abnormal proliferation of cells. Normal cells display a density-dependent inhibition in regards to cell proliferation. This means that normal cells will proliferate until they reach a finite cell density, then cease. The cell density is determined in part by growth factors, and when these growth factors are mutated, the cells will continue proliferating. So at a certain point, you only need so many skin cells on your right index finger. And once it reaches that, it has hit the finite cell density. But if there's something wrong with the growth factors in just one of those cells, the cells will continue proliferating. They'll continue making new cells that don't necessarily need to be there. And this is where we really talk about cancer. There are more than 100 distinct types of cancer, with only a few occurring frequently. So there are a few that we hear about a lot. The four most common cancers are breast, prostate, lung, and colon rectum. I've heard about these a lot. I'm sure you have too. And that's why I'm bringing them up. Because when these cells begin to proliferate and clump together, they form a tumor. We've heard that word before. This is a good image of a lung tumor. Tumors can be benign or malignant and will sometimes metastasize to other parts of your body. So this is when these cells have gone out of control and they've clumped together to make this tumor. If it's a malignant tumor, there is every chance that it can metastasize and move to other parts of your body. Classification of tumors will depend on what type of cell they arise from. Most cancers fall into one of the three main groups, which are carcinomas, sarcomas, and leukemias or lymphomas. Tumors are also classified according to the tissue of origin, an example being lung carcinoma, which this is every chance that is an example of a lung carcinoma right there. So these cells have been proliferating for a long time. That's a pretty good sized tumor right there. But see, this is what happens when the cell cycle goes unregulated. So let's talk about cancer and cell cycle disruption. Cancer cells are cells that have gone wrong. They have lost their way. They have mutated. Something's gone where instead of going to the GO phase and stopping, they just continue going and going and going. They originate in tissues of the body, but they can diverge. Their original cells and tissue that diverge from normalcy and kind of go off on their own. Mutations are the most common cause of cancer. These accumulate in genes that control proliferation. So if there is just a small mutation in the gene, that the growth factor of a cell per se, and it never tells the cell to stop growing and making copies, it will continue on and on and on. Different cancers have different mutational signatures. Not all cancers have the same mutation or will have a mutation that's in the same place as a different cancer. So that's what makes cancer harder to to treat and to study, is there are so many different mutational signatures. How cancer spreads is they have proteases that they secrete. These are enzymes that kind of go in and they will degrade that extracellular matrix, creating new pathways for these cells to venture out into the rest of the body. Now, these are cells that were going through a normal round of mitosis, but there was a problem, an anomaly, and instead of finishing the cell cycle in a regulated way, they are now cancer cells that are continuing to move on and move into the body and wreak havoc. So that is our lesson. I hope everybody enjoyed it. Thank you for joining me today. I look forward to some very interesting discussions, and have a great night.